of the gene pool of the whole population. So they're going to start probably growing. They can go extinct, but they can start growing a new population. But then it's just like a subset of the population that from they came. So this called the one that I graph here, they call like the founder, and then the founder effect. So it's going to have low gene diversity, and it's going to have low population, and because it comes from a big population, but it was just few individuals that moved to a different area.
So then over time you can distinguish those two populations, right? Because only some of the alleles moved into the new place, and those are the alleles, that's what the founder effect is, right? Those are the alleles that will then spread throughout that population. But now you have two distinct populations because the original area had a whole bunch of alleles, and that founder only represented a certain number of them.
And if, for example, as the paper was saying, from Africa to Europe and then to Asia was probably some individuals of this population that moved here. So again, it's like another founder effect, and then you can actually track back all the way to the original population.
So if this is confusing, which it easily is, right? Wikipedia's entry on most recent common ancestor is actually really good. Okay, so even if you just Google the acronym they use over and over in the paper, which is TMRCA, MRCA stands for most recent common ancestor. Their entry is actually pretty good, especially the part, and I wrote this down so you guys would know, the TMRCA via genetic markers portion of the Wikipedia entry is a nice way of explaining how genetic markers are used to calculate what was just discussed here. Okay, so you probably have learned this at some point. You may have forgotten it. You may have a textbook that can help you too. But that Wikipedia entry was actually, in terms of Wikipedia entries, pretty good. So I would suggest, if the whole premise of this paper was confusing, that's a good place to start.
So in the paper, so this paper doesn't have different sections as a regular paper, and that's basically the format that the science and nature have. But just roughly, you can see that the paper has two different sections. One, they do a simulation, and in the second one, they actually test some of the parameters with real data. And that's when they actually include the human genomes that they have from Africa, from Asia, and from Europe. Was that clear for you in the paper?
Like figure 2 versus figure 3, right? So if we're looking at that, on the left is the simulated data, and what that means is they made it up. Like they just put certain parameters for the alleles into the computer and said, show me what my program will give me back, and let me see if it matches the information I put in. And on the right-hand side, you have real data put into that same computer program. Okay. And you do that to check the efficacy of your program, right? If you know what you're putting in should give you something, a certain result, then you can see whether or not it does. And so they found the fallacies of their program, and they spend a big chunk of the paper discussing that, right? Like discussing the fact that if you're too close to now or too far away from now, the program doesn't work so well. And that's somewhere on the first page. The right-hand side of the first page, they discuss that, right?
So the results that they obtain, because as she was saying, so you can put some parameters and you know what to expect, and then you're expecting the program to give you what you want. And then they have this time intervals. So they knew that the results were not accurate. For recent times, they were not really accurate. For really old times, but they were really accurate, in the middle. And so based on that, they were actually putting some real data. They know how the confidence of the program is, and which, time window is the confidence. And then they were putting some real data to actually estimate some of the population parameters like this. All the descendants of the gene flow between different populations and the different migrations in human populations.
Could you go over the PSM model, ASMC model?
So, well, that is kind of complicated. So, let me see. I think I have a chart of that. Let me see if I can find it.
Okay, while she looks for that, can anyone tell me what they used to use for this type, prior to this paper, what they used to look at this type of data, right? So they had information they could put in, a certain type of DNA, a very specific type of maternal DNA, mitochondrial, exactly, right? And so that's the first part of the paper, is it sets up why they would go into this. Like, why do we care, right? So they used to use mitochondrial DNA, but they give you the cutoff for when that can be useful, which is about 200,000 years ago. Before 200,000 years ago, when you're trying to look at differences, you can't see anything earlier than 200,000 years ago with mitochondrial DNA. So what they're trying to use here is X chromosome and autosomal results, right? And again, if this is confusing to you, I highly suggest that Wikipedia page, because it actually talks about the mitochondrial DNA as the basis for its discussion, and then it kind of lets you see what the fallacies of that were and why this paper exists, pretty much.
So that is based on the coalescence principle. And so what you put into the program, because it's a program that develops an algorithm, you put all the allele frequencies that you have now on the population. So this is basically what you have now. And what you do is to trace back those alleles, to find the common ancestor of those alleles. So there's supposed to be one allele that is basically the ancestor of all the ones that you have right now. And so the program just looks from now back into the past until they find the common ancestor of those alleles. And so that's simplistically how that works. So, and that goes back to your original questions of, the founder effect. They have some alleles now in the population in Asia, some alleles in the populations in Europe and Africa. And then if you trace back those alleles all the way, you find where the ancestral allele was most possibly have arrived in general. Is that clear? Yeah.
So there's a certain mutational rate that has been calculated and that they use here, right? And they put that mutational rate underneath each of the charts in Figure 2, right? And if they look at the number of mutations, or the number of differences in those alleles in the now part of that chart, right? They look at the number of differences that exist there, and they use that plus the mutational rate to find out when these individuals had a common ancestor. All right? Now, if you look in the supplemental methods, you can see all the math they used to write that algorithm. I looked at the supplemental methods and went, I don't understand any of this, and had scrolled all the way down, basically, and then they were done, and I got nothing out of supplemental methods because that math is well beyond my abilities, right? But suffice it to say, if you flip through there and you're very good at math or writing algorithms, you're going to be able to understand where this algorithm came from. That's beyond the scope of this class. All you guys really need to understand is where this diagram is coming from, and the fact that it's the number of differences between the individuals that they're looking at now, plus that mutational rate. You combine those two things together, and that allows you to get the time to the last common ancestor.
So remember, you can go a little very deep in details and methods, but you don't have to for this seminar. You really need to get the big picture of it. So it's you need to put some of the methods that they use, but you don't really have to go into a lot of detail. It's more important the interpretation of or the results and how you interpret the results and what are why is this research relevant, for example. So this paper was there. This is one of the most important journals. It was there mainly because of the methods they're using. They're really, really new methods, and the fact that they are using the whole genome instead of using a few genes. A lot of people do this using a few genes. They're using the whole genome, basically the whole genetic structure of individuals. So basically, you're not missing anything. And then the technology that they're using and the software they're using to explain some patterns. And patterns, are kind of like going on back and forward, and humans have been going back and forth and understanding. So when the migration from Africa to Europe happened, and so if there was one migration, or how many migrations there were in this paper, because they were able to use the whole genome, they were able to answer better these questions, and they actually realized that there were some gene flow, some migration until like recently, and people thought about that for a while. They thought that it was just one migration, and then they were not connected at all. So using those new techniques, are able to bring those old questions and to actually give new results and new insights into what we know about human evolution.
Do you guys know how whole genome sequencing works? I'm not sure how much, so quite honestly, when I went to school, it didn't exist yet, so I don't know how much detail they go into with that. But do you guys know about aligning sequences and things like that? So there's a reference genome, right? And so they use the human reference genome here because we're looking at human DNA. And then you do Illumina is the technology they used here. So you do whole genome sequencing where you prepare, in the case of the human, it must be a decent number of samples, and you run them on lanes basically, and you get back from the Illumina computer a whole bunch of short reads, so little chunks of DNA, right? And then what you have to do is you have to use a computer program to line up those little chunks. You look for places where they overlap, right? And so what you end up with is a reference genome. So if this is your reference genome here, right? Then you have millions and millions of reads that get aligned. And so one might be here, and one's here, and one's here, but then you're going to get one here, and one here, and one here, and one here, right? So you get this overlapping sequence that allows you to put all of these genes in order. So you can see where they would fit on a single chromosome. Does that make sense? You need this reference genome to line up those little tiny pieces you're getting so that you can know what order these genes go in. So you can look for alleles or differences between two individuals because you know you're looking in the same place. Okay? So that's what's super important about doing alignment correctly. This paper takes for granted, they give you the reference for the alignment tool they use, but they take for granted the fact that you understand how the alignment happens in the first place. And so that's basically a very simplistic view of how it occurs. So you get a gajillion of those sequences back, and you send them into a supercomputer, usually, or it takes a really long time if you're running it on a laptop like I did with the Arabidopsis sequencing. I'd type something in, go wait for 45 minutes, come back, and like a little part of one chromosome would be done, basically. So you just have to be patient or have a really good computer, and it lines it all up, and it spits out what your overall sequence is for that single organism you've been looking at. Does that help put this paper into context a little bit more?
So besides the methods, did you understand that you follow the results that they obtain about the different migrations and the relationships between humans from Asia and Europe and Africa? Was that clear from the paper?
No. We have at least one no. So, oh, you go. No. You go.
That was going to have somebody else. Somebody got it.
Yeah. Does anybody think they kind of understood at least the one major result that was there? Kind of in the last couple paragraphs. So does their data go along with the commonly published data, or does it fall more along the anthropological lines in terms of when the African, European split occurred, or African, non-African split. So, right at the end of this paragraph here, on the last page, right here, there's a sentence that basically ends with the idea that their data says that that African, non-African split happened not 60,000 years ago, as is the commonly published date, but was much closer to now than that, was more 20 to 40,000 years ago. And there's also some anthropological data that is published elsewhere that goes along with that. So they're not saying something that's like completely out of left field. They're just saying genetics now map to this stuff, this idea that anthropologists have had for a little while. That maybe even after the bottlenecks that are pretty obvious, there was still genetic crossover between those two populations.
So for the essay, there are, as I mentioned before, two main results, and one is related to the simulations that they did and what they proved with the simulations, and then the other is, putting that into real data. And with the real data, that's one major result, what she mentioned. But there are also some results comparing, populations from Asia and Europe. That is also an important result for the paper. And then for the discussion, you need to go beyond what they say. You need to talk about the relevance of the study. Why is this important? What things are bringing new to our knowledge? And go beyond. So they spend a while in the last paragraph probably talking about the problems and the data still is not accurate. So what's beyond that? So what are the future directions of research like that?
And also when you talk about the data, for instance, let's say from the simulation, right? So one of the things, if you're writing an essay especially, although doing this in the worksheet is also important, one of the things that we look for is whether or not there's any sort of tie in or fluidity to your essay, right? If in the discussion you're discussing the results that you say came out of the purpose, right? So if you say what the purpose is, and then you say, here's the methods to get these results are interesting because of this, right? So there's a place, because there are a lot of caveats in this paper. There are a lot of places where they say, this wasn't quite right, with the simulation, for instance. That's a result, right? The fact that they did not get back exactly what they put in. That's an important result, that it wasn't exactly right. And in the discussion, you can discuss what you think that means in terms of the power of this algorithm. Right? So trying to kind of tie things in together is a good direction to take. Those of you who printed this out in black and white and then attempted to read it in black and white, yeah, you need the color for this guy, okay? So, if you tried to read it in black and white and you didn't have a computer available, go back and look at the figures on the computer, because it will enlighten you to what is going on quite a bit. That happened to me. It was much clearer when I could see which color they were discussing and what was going on.
Any questions about any methods or anything from the paper?
only have one more week of papers, and then we're back to seminars. So if you like the seminars better, we have one more paper coming up next week. And then it'll actually be job talk seminars, which I told you guys about at our last seminar at the end. So I think those are going to be, please God, really helpful to you guys. If they're done well, it's a perfect example of laying out, here's what I was looking at, here are the results I got, here's what I want to do next, right? So hopefully those will be good. Okay, I need you guys to sign in, and also if